Hello. Welcome to the tutorial on the Fungal Cytochrome P450 Database. The database allows the user to carry out comprehensive analyses of cytochrome P450 proteins, hereafter referred to as CYPs, in fungal species. Currently, the database stores a total of 22,940 CYPs from 213 species. Out of this total, 9,397 CYPs are from 113 fungal species, and we have included 13,543 CYPs from 100 non-fungal species so as to facilitate comparisons. The user can search the database from the welcome screen using the CYP protein ID via the search function, as well as by using the P450 name mentioned in the FCPD system. The guide to the P450 database takes the user to the current tutorial video. Updates to the database can be viewed in the News and Notice section. The database consists of 7 main pages that allow the user to analyze and understand the CYPs stored in the database. These 7 sections will be introduced in the respective tutorial videos. The Species tab allows you to explore CYPs belonging to various species. The Species table under the Species tab displays information regarding kingdom name, species name, the number of CYPs in the genome, and the total number of the open reading frames, also known as ORFs, encoded by the genome. Upon clicking the CYP numbers, you will be redirected to a list of CYPs on a new page. The CYP list page, as its name suggests, lists the CYPs encoded in the genome. The table displays protein IDs, the best blast hit to the CYP from Nelson's database, the species name, and the interpro domain class to which the protein sequence belongs. As a user, you can click on any protein ID and retrieve the sequence. Going back to the Species tab, the CYP list page features 2 additional tabs. The Introduction tab gives you any additional information regarding the species. The CYP Name tab gives you a summary of the CYP family representation in the species. The Class tab. The Class tab displays the distribution of CYPs into the 16 interpro classes. The table also shows hits to CYPs from Nelson's database. Looking to the Class Species Matrix feature. The Class Species Matrix link redirects the user to a page that displays the distribution of CYPs into 16 classes for each species. For a given species in the database, the species may have genes represented in a few of the 16 classes, or several of the 16 classes, but not necessarily in all 16 classes. As an example, when viewing the initial list of species on the Class Species Matrix page, Phytophthora infestans, the notorious oomycete responsible for the Irish potato famine, possesses CYPs in 5 of the 16 classes. The Cluster tab. The Cluster tab displays a searchable list of the clusters generated by the clustering procedure. The Cluster tab shows the cluster number, the number of fungal CYPs in each cluster, and the total number of CYPs in each cluster. Each cluster leads to individual pages that list the CYPs in that cluster, and any CYP family represented in the cluster. The sequences can be obtained by clicking on the protein IDs. The CYP Family tab. The CYP Family tab includes CYP family distributions across the fungal phyla, and, for broad comparison amongst eukaryotes and prokaryotes, representative species amongst non-fungal phyla. The SIP Family tab also displays the clan distribution and includes the number of clusters corresponding to each family. Specific SIP families can be accessed using the search function. For example, SIP 560 results are displayed as shown. The SIP Clan tab. The SIP Clan tab shows the SIP clans in fungi. The table consists of clans and the distribution of SIP families within these clans. The clans and families are linked to SIPs belonging to them. Clicking on the clan or family redirects you to the page with SIPs. For example, when clicking on SIP 5016, the user will be taken to a page where the user can view all associated SIPs. Or, for SIP 5016, the user can choose to view only those SIPs associated with metazoa, which can be done by clicking next to metazoa and then hitting apply. These SIPs can be viewed by selecting specific phyla or subphyla. The sequences can be accessed by clicking on the protein IDs. Going back to the main SIP Clan tab. At the end of the table are orphan SIP clans. Also listed are Nelson's SIP families that do not have homologs to SIPs found in the database. The Characterized SIPs tab. For putative functional annotation, we collected characterized SIPs and compared them against our database. This tab stores the results of that comparison. The characterized SIPs are divided into putative functional categories: primary metabolism, secondary metabolism, and, on subsequent pages, xenobiotic metabolism. The homologs in the database to each of the characterized SIPs can be accessed by clicking on the number of hits. The page includes the SIP hits and the SIP family corresponding to that hit. Going back to the main Characterized SIPs tab. At the bottom of the Characterized SIPs page, users will see the new P450 feature. This feature allows users to enter the sequence information for newly characterized P450s. After clicking on the new P450 button, the user will be redirected to a page where the user can enter details, such as the group name, the NCBI accession number, the description, if available, the species name, the amino acid sequence, and the E-value for comparison. After clicking the Submit button, users will be redirected to a page which resembles the original Characterized SIPs page, but will display the newly characterized P450 data alongside the previous characterized SIP data. The BLAST tab. The BLAST utility allows you to search the database with your query sequence. The user can define parameters for the BLAST search. The BLAST utility allows for annotation of putative SIP sequences. The user can copy a fungal nucleotide or protein sequence using the BLASTX or BLASTP options, respectively. For instance, for the filamentous fungal pathogen Aspergillus nigilans, the user can paste the sequence into the box and click Submit to retrieve the BLAST results. These BLAST results resemble the output from the standard NCBI BLAST output. Storing sequences into favorites. The FCPD 1.2 database allows users to collect one or more sequences and store them into favorites. This can be done by checking each sequence, or all of the sequences, and then, at the bottom of the page, the user can click the Browser button and create a favorite. Once a favorite is created, the user can store the sequences into the favorite by clicking the Save button. Once the sequences are stored in the favorite, the Go button will take the user to the Comparative Fungal Genomics platform. In the CFGP, the user can select sequences in the favorite and carry out analyses.